So I've got a few relevant disclosures. I am trialling quite a few vaccines at the moment. I do provide some advice to AstraZeneca, but of course these views are all my own. And the outline's going to look pretty intense, and the slides, some of them are a bit content heavy, but I'll just focus on the key bits, so don't try and read too much of the stuff that's in the slides, and as I say, we'll leave plenty of time for questions at the end. So, obviously if you believe social media, there's around 2 million infectious diseases experts and epidemiologists in every state of Australia at the moment, but there's a few of us that do get asked to do quite a bit of media, and I just thought I'd point out some of the reasons why. Hopefully it's in some ways justified that they chat to me a bit. So as you heard, I do work in infectious diseases and am a microbiologist, so involved in some of the testing, but I do work closely with clinical trials, and particularly in new vaccines, and have done so for a while. So at the moment I'm directly involved in around 10% of the world's COVID-19 vaccines, particularly the early phase trials, and I'm also part of another group called the Immunisation Coalition at the bottom there, and I've just included a little flyer. We had a media release yesterday where we're trying to advocate for paying forward, given we get the vaccines for free in this country, and there's a lot of third world countries that still don't have access to vaccines. So we've got a little campaign we've got out there at the moment, and there's a flyer in your tables about that. I won't dwell on the basic stats too much, but the picture on the bottom right there is a little cartoon about putting pandemics in context, and COVID-19 is ninth, but rapidly climbing up that scale. And as people will be aware, we've been very fortunate in this country to have exceptional control with around 30,000 cases and still less than 1,000 deaths, which is remarkable in the global context. I just thought I'd flash up a list of all the available specific antiviral therapies that we have in use at the moment. So there's still none. So this is what underpins why it's so important that we focus on prevention, and obviously the best way of doing that is with a safe and effective vaccine rolled out to everybody who's vulnerable. So in terms of the history of coronaviruses, they've been around since the 60s, I guess like a few people in this room, the name comes from Crown, so it's nothing to do with the beer, it's how they look under a microscope. And this, obviously everyone's talking about coronaviruses at the moment, but this isn't the first one that we've had big problems with. We had the first SARS back in 2003, and it's important to remember that event because that's really what started vaccine development for this group of viruses, and why we've been able to do that so quickly. So I'll just touch on that as well. A lot of people are hotly debating where this came from, and this is something that comes and goes in the media all the time and has undergone a bit of a resurgence in the last few days and weeks. We know lots of viruses we've had to battle over the years have come from animals, particularly bats seem to be very good at doing that, and there's a list of those there. The first SARS in 2003 did come from horseshoe bats or palm civets, while this virus was first found in that wet market, looking really closely at the distribution of the people infected in that market would seem to indicate it was brought into that market, and that was just where it was, the spread was facilitated given the close proximity. A lot of people are talking about an accidental or intentional leak from that lab, and there's this gain of function research where people were able to show they could change these viruses to be more transmissible, but the people doing that would have absolutely known what the potential for release was there, and I'm sure wouldn't have done that accidentally. And there was a very similar, so about 96.2% homologous virus found in the feces of bats as far back as 2013. So it would seem very likely there was a similar virus circulating in bats and or other animals, and we've seen it evolve since we've discovered this virus. We've seen it change and have these variants of concern. So that evolution perhaps started in whatever animal reservoir it was to be able to make that jump across to people. And we call that a spillover event, and just to show some of the spillover, the viruses with spillover potential, there's a big long list there. The top 10 on that list are already viruses that have shown they can infect people, but you see that the next 4 of those are coronaviruses. So they are a virus that we think has the potential to make that jump, and there's quite a few other coronaviruses that are being monitored very carefully for indeed the same thing to happen. And so, as I said, coronavirus vaccines didn't just start at the start of last year. We had lots of really good candidates. We've got some good ones for animals, and I've listed the couple up there. And we had lots of really good candidates for that first SARS, but that was a very different sort of infection, much more severe, not quite as transmissible. And so because it was more severe, people that had that were sick, they were in hospital, a lot of them died, but we were able to eradicate that with the basic infection control principles that we're still applying to this. So we only had about 9,000 cases. And so all those vaccines that looked really promising, those first trials, couldn't make it to phase 2 and phase 3 trials because there were no cases left. But a lot of the vaccines we're seeing now were born out of that research. So when people ask how do we do it so quickly, a lot of it is because it's based on 20 years of research, not just a few months. In terms of how quickly we're able to make these vaccines, a lot of people are sceptical because it was very quick, but there's a few reasons we're able to do that. One of the main ones was funding. So we had more funding than ever before. There's a group I've listed there called CEPI, the Coalition for Epidemic Preparedness Innovations, and they already had a funding campaign for a response just like this. And with that UQ vaccine people would have heard about, we secured some funding about 3 years earlier to be able to have a program to respond to what they call Disease X. So when this sort of thing happened, we'd be able to really rapidly scale up a new vaccine. And unfortunately, we had some plans to do some practice runs, but never got to do the practices because this came so early. But that was what underpinned that program as well. So funding was a big player there. As I said, we had lots of previous research that these vaccines were built upon. And because of that funding, we were able to do a lot of steps we normally have to do one after the other at the same time. And the big one there was scaling of manufacturing, which is a huge gamble, costs an enormous amount of money. But all the lead players, particularly Novavax, for example, started their scaling of manufacturing at the start of last year in parallel with the clinical trial. So if that vaccine hadn't progressed as well as it did, obviously that money would have been wasted. But it means now that they're just about to finish the phase 3 trials, they're ready to make billions of doses to roll that vaccine out fairly quickly. And also some of the things about study design and getting almost real-time approvals from ethics was another way we could expedite some of those trials. So at the moment there's 93 vaccines in clinical trials for COVID. This tracker here obviously adds up to a lot more than that because some of them are still completing the phase 1 but all the way through to being approved or unfortunately abandoned. So we have lots of new vaccines coming through. And I just thought I'd touch very quickly on the mechanism, again not to dwell too much on the science, but just so people I guess understand the key differences. So a lot of people are talking about nucleic acid vaccines, and again this is something that has been around in principle for quite a long time. Moderna is a company that's been around for over 10 years. And what this is, so this is the Pfizer vaccine and Moderna we're going to get later, is in the end all these vaccines, what we want to do is train the immune system to be ready to respond to the virus when we ever encounter it. And we often do that by exposing it to something on the surface of the virus, and in this case it's the spike protein or S protein, the thing on the surface. And with the mRNA vaccines, what we do is we basically inject the instructions to make those spike proteins. Unfortunately, that's something that's worked really well. So we've seen with Pfizer and Moderna, that's an incredibly effective strategy. And those little bits of mRNA also really help to stimulate the immune system. So that's why we've had such a potent response. One of the main questions we get asked about that is because it sounds a bit like DNA, does it incorporate and change people's DNA? And there's absolutely no way it does that. So that's a fallacy that simply isn't true. The viral vector vaccine, so this is what AstraZeneca is. So again, we want to get those spike proteins, but in this case, instead of using the instructions in the form of mRNA, we put the instructions in a harmless virus. So a vector virus that that takes that into where it needs to be. And in the case of AstraZeneca, it's an adenovirus, a chimpanzee adenovirus, and some of the other ones, it's a benign human adenovirus. So again, delivers the instructions to make those spike proteins. And again, we've seen that's a very effective strategy. Again, the fact that virus is there helps to really boost up the immune system. It does also make it a little bit what we would term reactogenic, so people often do get a few side effects with the first dose particularly, those things like fevers and aches and pains and that sort of stuff. And I'll talk about the TTS, that rare clotting thing, to hopefully put that in perspective in a little bit. The protein ones is a much more simple and traditional way of making a vaccine, so that's where we just make those little bits in the lab and inject that in. Trouble with those is and Novavax is a good example, as was the UQ vaccine, and there are some more of those coming through. In its simplest form, if we do that, we just don't get enough of a response. So we have to do a few clever things to make that work. Usually we have to try and do something to keep it in the right shape, because those proteins can stick together or fold into the wrong shape. In UQ, that was that molecular clamp, that was what that was all about. And with Novavax, they make them into nanoparticles. And when I say nanoparticles in the media, a lot of people think that means microchips or some other sinister, silly thing, but there's no microchips involved. It just means it looks like little bits of virus. That's what they do. And then we use another thing to boost the immune response called an adjuvant. So, fortunately, we'll see Novavax hopefully come online in this country fairly soon. The final way we have of making these vaccines is just to take the whole virus and do something to it so it doesn't cause disease. This has been something that's very popular in some Asian countries, but all of these haven't really shown enough efficacy that we'll be considering them in our country, so I'll skip over that. A lot of people like to compare these vaccines. So a lot of people are saying, and for some reason Pfizer just seems to be the trendy or the popular one at the moment that all the cool kids want to get, and AstraZeneca is the rubbish one. And again, I've got a slide here comparing a few key things. If we look at that second row, the main thing is prevention of severe disease is almost 100%, which is just astonishing. And if we look at no other properties of these vaccines, that in itself would be enough to justify using them. In terms of comparing AstraZeneca and Pfizer, I've got a heap of text here. Don't read that. The key thing is Pfizer better than AstraZeneca? No, it's not. And the simple thing is we haven't done head-to-head studies, but when you actually look at the real-world experience, so these vaccines have been used in hundreds of millions of people now, the AstraZeneca vaccine performs basically the same as Pfizer. So one is not better than the other, particularly in terms of how effective they are. So adverse effects. So a lot of people do want to talk about what these vaccines do. So in the clinical trials, it was really clear they did cause some side effects. The big thing about the numbers we get from clinical trials is we were asking those people every day. So the day after you get a vaccine, we call you up or get you to type into an app, was your arm sore? So very few people are going to go, absolutely not, I have zero pain in my arm. So we get a slightly inflated perspective of adverse events from the clinical trials. So that's why we need to monitor those things moving forward, which we do. So as with all vaccines, a lot of people get a bit of a sore arm, nothing of concern there. And with these vaccines, as I said, because they do generate such a good immune response, they're a little bit relatively reactogenic. So more than you'd get, say, with your flu vaccine, people have some aches and pains, maybe get a fever for the first 24 hours. But rarely does that mean you can't go on with your day-to-day activities. But we do monitor these things really carefully, and I'll come to the TTS stuff in a second. You know, that demonstrates a strength of how closely we monitor how these vaccines are performing in use, not only in our country, so this is some TGA data, but also in the whole world. So obviously headache is the most common thing people report, but I can't see the numbers. I think it's about 6 per 1,000 doses or something like that. So it's still, the incidence is still relatively low. So we do expect some people to get some of those things, but it certainly shouldn't change people's perspective of those vaccines. I will come to that clotting thing now because so many people want to talk about it, and there's so much misinformation out there at the moment. So has everyone heard of TTS? So that rare clotting syndrome has changed names about 18 times since it was first discovered, probably only about 6 or 8 weeks ago. In fact, so VIPIT, VTTS, that, and all sorts of things. And one of the big things to say here is this is not the same mechanism as normal clots. So most people know someone who's had a DVT or a PE after an operation or a long haul flight. And we get a lot of people saying, I had one of those clots, or my auntie had one of those clots 40 years ago, I shouldn't get AstraZeneca. That's a very different mechanism. So this mechanism increasingly seems to be driven by the immune response to the vaccine. And we're not exactly sure why. Lots of good work is happening to try and work that out. But it does seem to be driven by an immune response that unfortunately means your clotting cells, your platelets are low, but you do get clots a little paradoxically. It's not something that happens in the first day or two. So if people have a headache the day after the vaccine, that's not this thing. It usually starts between day 4 and day 20, out to maybe day 28 now, but usually between day 4 and day 20. And usually the symptoms are not subtle. So, and to highlight again, it's very rare, but in people that do get it, you kind of know about it. But one of the things that's really important in this country is we're so good at looking for it. So we're very confident we're not missing any of these, because anyone who comes in with any side effects gets blood tests and scans and taken very seriously. So we have a really good algorithm for looking for this, for not only finding it, but confirming it in our country. And so our management has been much better as well. So early on, we were really worried it was quite severe, and mortality figures were quoted at 20, 25, 30%. But as people probably know, we've had 2 unfortunate deaths in this country from 60 reported cases, meaning our mortality overall is probably going to be less than 5%. So this rare clotting thing, while obviously significant and it has affected how we use that vaccine, is not a death sentence in itself. Most people will potentially need to take a blood thinning tablet for 3 months and recover fully. So I think a lot of people don't yet have that sort of thought in context. And again, not to read all the text in this, I know it's a very busy slide, but what we have to do throughout is weigh up the risks and benefits. And this happens with all vaccines, with all medicines, and not only happens at the approval stage, but happens on an ongoing basis. So this is what a TAGI, the experts that advise the TGA, do all the time. So they look at the pros and cons cons. And you can see the list of positives on the left there is far longer than the negatives. And there's a few really important things to point out. AstraZeneca is an effective vaccine. We know it works really well. We know we have supply issues with the alternate vaccine, so we can't just replace it with Pfizer. A lot of people argue, well, we have no cases in Australia right now. Why would I bother getting vaccinated? But, you know, events of the recent weeks, Victoria, New South Wales, and now locally would highlight the fact that we can't keep this virus out forever. And one of these days, we're going to have to start to allow some travel and some opening up. And when we do, the virus will come back. And we've seen that in Taiwan and Singapore and other countries who had perhaps even better control than us at the outset. So we do need people to be vaccinated, and there's a big risk in not being vaccinated. And that is that we have to rely on lockdowns, border restrictions, and all the stuff we're seeing now. So the way out of that is to have enough people vaccinated. And the way for us to do that is to use AstraZeneca. Versus obviously the negative, which is incidence rate overall is probably 1 in 100,000, maybe up to 2, with, as I said, a mortality of less than 5%. So we do need to weigh those things up. And that's what ATAGI are doing on a constant basis. Just some things to put it into context. So those sort of standard clots that we talk about as a comparator are very common. So about 50 people per day get those sort of standard clots in Australia. What I think has been a real challenge here is people have lost a bit of perspective. So our annual road toll is over 1,000, but unfortunately when people succumb from a traffic accident, it doesn't make the front page. But every even suspected case of TTS makes every media outlet in the country. So I think we've got an inflated perspective of how common this is and how severe it is. So even a lot of those cases you hear about then go on to be not confirmed as related, but there's no front page thing 3 days later to say, oh, actually it was caused by something else. So I think we've got an inflated perspective of just how common this is occurring at the moment. And just to sort of highlight that in a few pictures and to put some things in context, the reason we've made an age change is not because we value people over 60 any less, as a lot of people are asking. Why don't you care about us? Is the risk of the clotting thing goes down as you get older, so it's safer, but the risk of severe consequences from COVID goes up significantly. So overall, if you weigh up the risks and benefits of that vaccine, the benefits still outweigh the risks, but that gap is greater as you get older. And so out of an abundance of caution, that's why that age cutoff of 60 was brought in. But just to keep things in perspective, we know things like smoking increase the risk. We know that clotting in the actual COVID infection itself is very high. But just, I don't know if anyone rode their bike here today, the risk of dying riding your bike is about 1 in 140,000. The risk of running, about 1 in a million. And I even found an article that showed that an increased risk of dying sitting on the couch watching TV. So if anyone's going to binge watch anything on Netflix, you're potentially adversely impacting your mortality more than receiving an AstraZeneca vaccine. So just to quickly touch on some local experience, as I said, I've trialled quite a few vaccines, and one of those was Novavax. So this was the first vaccine trialled in human volunteers in the Southern Hemisphere. A really interesting product. I've worked with Novavax on about 6 previous vaccines. Unfortunately, none of those ever made it to market, but their COVID candidate is looking exceptional. Some data released at the start of last week shows maybe up to 96% effective. They've had some issues with manufacturing due to supply of consumables and some challenges getting through their clinical trials, but with the data they've generated, I suspect they'll get some approvals in other countries any day almost. And so that is one we have an agreement to get 51 million doses of in this country, so will be a big part of our strategy moving forward, hopefully by the end of the year. I was very involved with the UQ candidate, and I won't dwell on that too much. Did look to be effective in the phase 1 trial, but had that unfortunate issue where that molecular clamp, which is the red bit at the bottom, as I said earlier, is the bit to keep the vaccine in the right shape so it doesn't stick together. And the protein that was used was one that was made from HIV, so no HIV in that candidate. And that GP41 protein was chosen because it had all the right properties and was a well-studied protein. And it was thought that the bit that we used for testing was kind of hidden on the inside, so we wouldn't get the issue that we did actually get. So, and that was thought to be too great an issue to overcome to use that vaccine. So unfortunately that one has been abandoned. I'm doing some work with one of the largest vaccine manufacturers, the Serum Institute of India. They have a really interesting candidate that instead of targeting that whole spike protein, there's this specific for what we call the receptor binding domain. And the thing about that is that specific part, so that's the bit that attaches to the receptors in people, is that's a bit less susceptible to changing as a variant of concern, I guess. So this vaccine might be one that doesn't have the same reduction in efficacy with some of these new variants. So if that works, that'll be great. Trialling an oral vaccine, which is really fascinating as well. So you recall earlier I said with the viral vectored vaccines, we put those instructions to make the spike protein in a harmless virus and inject that. Well, in this case, the same instructions have been put basically in a probiotic bacteria. So if you have your Valia in the morning, we've got essentially a Valia that's got a little bit that makes that spike protein. And so if that works, the idea will be we get a good immune response from ingesting that. And it's stable at room temperature as well, so it doesn't need those fancy fridges we need with Pfizer. And we've just started a new clinical trial at the moment actually, so if anyone's keen to volunteer, you can look at the Nucleus website and maybe come along and participate in this clinical trial. So this is also a viral vectored vaccine. With most of them, they don't grow or replicate. But in this case, we allow this one to grow once, so we get a lot more of that spike protein produced, probably 100 times. So the idea there is we'll actually use that intranasally because we'll get enough made that we might get a response through the nose. And then that immune response will be better at hopefully blocking the virus getting in, which is of course what we'd really like. I'll skip through this stuff because I think I'm nearly at time. I did just want to go through some things. So, you know, COVID has taught us a lot. It's obviously impacted a lot of people, but I think there have been some things to take away. People are wanting to listen to people like me. Usually we're the boring people that bang on about hand washing and flu vaccines and no one cares what we have to say. But at the moment, people are interested and wanting to find out more about infectious diseases and epidemiology and vaccines. So I think that's good. I think we all know about social distancing. We all know about hand hygiene. And, you know, we've seen rates of all sorts of other viruses and infectious disease plummet as a result of that. So a strategy that we should hopefully continue moving forward. We've obviously seen lots of innovation, not only in vaccines, but in testing, in diagnostics, sequencing, you know, whole genome sequencing. We're mapping the evolution of this virus. We had a great talk a few months ago from someone with a rapid test here, Sean Parsons from Malone, for example. So, and just how we do research, we've shown we can do things a lot more effectively and efficiently. And I think the ability for us all to work together, some of the cooperation, collaboration has underpinned a lot of that. Just quickly to wrap up, a lot of people still think these vaccines don't block transmission. And a lot of that comes from the discussions we had early where people said, do these block transmission? And we had to say we don't know because we don't look for that in the clinical trials early on. It's something that's hard to prove. But now we have a lot of real world experience. We know they do, probably 50 to 66%, so not 100%, but we know they do reduce transmission. So there'll be a lot of chat today about how a vaccinated quarantine worker passed the virus from one person to another in the hotel quarantine system, but that person wasn't infected. The thought is they had it on them somehow, which again begs the question how that can happen. But anyway, it's not that they got infected and passed it on. Somehow they carried it from one person to another. But we do know they block transmission. People ask about boosters. We will almost certainly need boosters. We don't know the interval yet because those clinical trials are still happening. Hopefully it's less than the gap is longer than every year, might be every 2 years or every 3 years. But one of the things we're already looking at is combining vaccines now. So I'm going to be doing a clinical trial probably with Novavax with their combined flu and COVID. And then so we might have 4 different strains of flu, maybe 1 or 2 different COVID ones, and then you get that every year, and so you can get protected and before you travel and all that sort of stuff. Variants of concern, I'll skip over, but we're going to see more of these, but people shouldn't be alarmed by it because we can change our vaccines to combat these. So there's a strategy in place. But still the biggest challenge, so having said all that stuff, is if people don't get the vaccine, this is what the future looks like. So we have lockdowns, border restrictions, and all that sort of stuff. The way to not have to do that is to have enough people vaccinated that the vulnerable people in our population are completely protected, and then we can get back to probably not where we were before, of course, but a sense of normality. And so again, I won't go through this in great detail because I think my time is up. But this virus will keep coming. I think everyone agrees now it's going to be endemic, so it's a virus we're just going to have circulating. But I think we've got the tools available, or we're very close to having the tools available to be able to control that and live with it. That's going to mean technology like apps and using those better in terms of how we check in, so our contact tracers can be on top of things. Obviously, the first generation vaccines, which we've got now, which are great, but we'll have next generation ones, and we might combine them. You might get a nasal one every 2 years and a new strain one every year or two. So we'll have a very good strategy in terms of vaccines. We'll get some therapies, but I think we need to start looking towards the future, because I think the strategy we're in now was always designed to be a temporary one to get us to the point we had safe and effective vaccines, which we clearly do. So I think we need to start making some inroads into opening up in a safe way soon. And I'll leave it there.